happens to know Dr. Zettler. She can't be here tonight, but she recommended him to come in and give the talk for us. He is a Hitchcock Professor of Biology at Illinois College. He's also a research associate at the Chicago Botanical Gardens, and he runs the Native American Conservation Center. Did I get that right? Sorry, so the Orchid Conservation Center.
I'm affiliated with it.
Affiliated with it through Illinois College there in his appointment. So we initially had said he was going to try and bring out some books that he just recently published called Saving Orchids. Unfortunately, he did have to get those from his publisher shipped out, and they didn't arrive in time. So if you do want his books, you'll have to order them online, and you can get them shipped to you through that. But he's a great resource. I'm hoping we're going to learn a lot about orchid ecology, biology, and conservation. So without further ado, I'll leave it with Dr. Zettler.
Thank you. Is this thing available?
You should be good to go.
Is that good? Can you hear me?
Yeah.
Okay. Thank you for the invitation. And I think I was here maybe 15 years ago to the society. The hometown society I belong to is the Prairie State Orchid Society in Springfield, Illinois. And Mitzi is, thanks to Mitzi who can't be here tonight. I teach 70% of the time, and this was a long week. My voice kind of gets softer as the week goes on. So if you can't hear me, by all means, show evidence that I'm speaking too softly. Can you hear me in the back? No? Okay. Thank you. I need the critique here. So I'll try to step it up a bit. So tonight, I'm going to kind of blend science with teaching. That's kind of what I do most of the year. And I do a lot of research in the summer months with my students who are undergraduates for the most part. So tonight, I'm going to give you a little bit of a whirlwind tour to how the orchid functions from a biological and ecological standpoint. And how do we conserve these incredibly special and unique plants? Because about half of the species worldwide are currently threatened with extinction, and it would be a shame if we lost what we have. So where did this come from? I grew up in north central Florida. As a child, I was the son of another Dr. Zettler, my father, who was an orchid virus expert, and he published several papers on the subject. He taught me a lot about the natural world, and growing up in the rural part of Florida, I was used to seeing all kinds of organisms shown here that when I go back, they don't really appear anymore, and I'm bothered by that. So my father kind of instilled in me an appreciation for nature, and that was galvanized by my upbringing in Florida. And there he is with one of the papers he published, Viruses of Orchids Under Control at the University of Florida. He had a postdoc named Raghavan Charudatan, who was my mentor. He hired me. He taught me how to isolate fungi in his laboratory. And between my classes, I would do that. That's how I learned fungi and how orchids eventually use fungi. And my dad worked with Tom Sheehan. Some of you know Tom Sheehan or heard of him, Tom and Marion Sheehan. He had a postdoc named Mike Kane. And it turns out that decades later, our paths have crossed, and Mike Kane and I worked with the ghost orchid quite a bit. So tonight we're going to hear a little bit about how this occurred and how it started. So I was a graduate student at Clemson. After I graduated from the University of Florida, I went to Clemson. And my professor said, you can study an orchid or a floristic study. Just map out the plants in this one area or the orchid. It was an easy call. So I chose the orchid, but here it is. So what do you do with it? Well, I used my fungus technique, isolation technique. I read a couple papers out of Canada, and I tried it, and it worked. It was the first time I was told by the authorities that this fungus, germinating the seeds of a native orchid that's threatened with extinction, it's beautiful. They germinated with the fungus I found in the roots, and there they are, little babies. And I was told this is called symbiotic germination. I was told that that was the first time it had ever been done in the United States. So off I went. This is what I do today. And I took my laboratory goodies, scalpels, alcohol burner, and I went to Illinois College. And it's the oldest college in Illinois that conferred a degree. Abraham Lincoln gave a speech here before he was president. And the science building is the new building next door to where he gave his talk. I met my wife here. She's a psychology professor, and I've been there for 30 years almost. And what I've done is, since I teach a lot, I wanted to have the students have an appreciation for nature like I learned from my dad. So what do? I give them a rare orchid seeds of a species that could go extinct in easily their lifetime. And I tell them, let's get a fungus, let's marry it with the seeds in the petri dish, and you can own this. Basically, it's your orchid now. So take good care of it, don't kill it. And this is what we do. We take undergraduate student learning, and they have an appreciation then for the natural world. And they learn how to do this. Very elaborate techniques that then can be used as a springboard to graduate schools. It gets them noticed more. But our specialty is growing these orchids with fungi. And we're going to talk a little bit about fungi tonight, but not a lot, to put you to sleep, I hope. So we had a daughter, trying to get her instilled in orchids. There's dendrobium in her hair. There she is. She's now a PhD student. Where does time go? At the University of Chicago. She's studying fossil mollusks in the Galapagos, it looks like. So we're very proud of our daughter, and I'm still working on her for orchids. It'll happen. But it's kind of sad because this is where I grew up as a child in north central Florida, and I took Audrey, my daughter, there with my dad, and none of those creatures, very few of them, are there today for her, and that really bothers me. So that's why I'm here. A long time ago, when I first started out, we used typewriters. And this is Randy Curra from Alberta, Canada. He was the expert of orchid fungi in the world at the time. And I found this fungus. I said, I think this is going to be the one that the orchid needs to germinate. And he wrote back, as far as I know, no one else on the continent has worked with the systematics of these fungi. And back in the day, 1989, a long time ago it seems now, this is kind of what was going on. We had the first GPS satellite placed into orbit. DNA fingerprinting was admissible as evidence in Florida. Microsoft Office was born. Email and internet were just becoming mainstream. A lot has changed. So since then, it's amazing the technology that we have. I don't have to explain that. And we're finding out that the historical record of the orchid family, you don't have to rely so much on fossils because there are very few fossils, the soft tissued things. But we now have some idea of when orchids first evolved and where they went, what happened, how did they get to be the largest plant family on Earth. We think sometime right after the Great Dying, the Permian extinction event, where about 96% of life vanished, probably because of volcanic activity around Siberia. This is when the first flowers started to appear on the Earth. And this is a winner. This thing, the first flowers that formed this time in history, changed the surface of the Earth. And it's still the group of plants that we have reigning today. Now, the one thing that orchids have obviously the flower, but there's something that they don't have that's in this drawing. And I don't know if you know what that is, audience. What do you think? This is where I have to educate you. Endosperm is what feeds the baby embryo in the flowers. And orchids don't have much of that at all, if any. So it begs the question, if this is a baby orchid embryo in a seed, surrounded by a food source, endosperm, If orchids don't have the endosperm, then what's feeding the embryo? Yeah, of course, thank you. And that's what we're going to talk about a little bit tonight. We also have a little bit of fossil evidence that bees, this is some amber, I believe it's about 30 million years old, amber. There's bees that were flying around with pollinia on the back of their bodies. So we have a little bit of fossil evidence. But bees came along when the first flowers were coming off, as in the record, followed by Lepidoptera, butterflies and moths. And they marched into the Cretaceous. This is about when the age of dinosaurs was reaching its peak. And then it abruptly ended 66 million years ago, probably because of an asteroid. But flowers were around when the dinosaurs were here. We think that about 83 million years ago, orchids as a crown group came about in Laurasia, and then off they went. This is about when the continents were beginning to drift apart. But 83 million years ago wasn't that long ago, if you think about it. Well, sorry. We got to think about the big picture now. And as it turns out, the orchid family, it might be counterintuitive, it's one of the 15 oldest angiosperm flowering plant families. So, wow, I didn't know that until recently. It's now considered to be, now if you like the Asteraceae family, the asters, you might argue with me, but I can argue back, that the orchid is probably the most diverse plant family with roughly 31, I think it's now 31,000 species, 700, 800 genera. It's debatable. 70% of the orchids today, of that total, live in trees as epiphytes, like cattleyas and epidendrums. They've learned, they started out on the ground and marched into the trees and went boom and covered the earth, especially where it's warm and cloud misty forest with moisture. They like that. And one particular family, the epidendrum group, is the one that really has the most diversity. So if we look at the butterflies and moths and when they evolved, this is an interesting slide. If you look at the very top, right about that mark in time, about the time that the first flowers were beginning to show up, the first moths came along to develop a proboscis, like a straw-like mouth, to sip liquid diets. So it's interesting that flowers and the first moths were small, we think, and they had mandibles, but then they shifted to a tube. So flowers with nectar and tubes coincided a long time ago. And as the moths appeared, the butterflies followed suit. And down here, at the end of the Cretaceous or so, about the time when orchids started to really take off, we see sphinx moths, large Saturniid moths, the Cecropia, the Polyphemus, the Luna moth. They kind of forked, but the sphinx moths are some of the pollinators of orchids today, like the ghost orchid. And it's interesting that during the past 5 million years, this is when the orchid diversity really took off. So that's rather recent. Now that's recent. You can say 83 million years isn't, but that's pretty recent. And it's interesting also that 7 out of the 10 most diverse places on Earth where orchids are found are in the New World, especially when South America was connected with North America through Central America here. This is where a lot of the species met. And I would say Colombia and Ecuador are generally regarded as the most orchid-rich place on Earth. In the Old World, you have to go to New Guinea. So most orchids in the world, species, are in this side of in this hemisphere. So in Ecuador, they're very proud to say that they have about 4,000 species. How many do we have in the United States and Canada? Well, top right. That really Ecuador is a small country. It's elevation, but there's only 250-ish species in the US and Canada combined. About half that number is in Florida. Anyway, it's extremely rich. But the downside is the orchid family is also the most vulnerable to extinction, because what people have been saying is they're canaries of the coal mine, meaning that they're the first to disappear when the environment changes, even subtly, because they're so sensitive to acute changes, the slightest change, and they begin to disappear. And that's a red flag, because if you're studying the ecology, you can tell that there's something up. They're disappearing. And here are some examples of what we would have to lose if we're not careful. Dracula from Ecuador. What can I say? I don't know if you like it or not. I don't know. It's very odd. We have Dendrophylax. This is the ghost orchid of Florida's genus. There's roughly 13, maybe 14 species in the Caribbean, Cuba. Cayman Islands has one, Fawcettii. We have the chocolate orchid. Some of you may have smelled this before. This was photographed in Cuba. Does smell like chocolate. If you blindfold yourself, I couldn't tell the difference. Inectocylus in Hawaii. Hawaii has only 3 native orchids, and this is, I think, is the loveliest, but it's got a modest appeal to it. And then there's one in Australia that is completely under the ground, pretty much the whole time. It was discovered when they were plowing a field. A farmer found this glob, and he dusted off the sand. He looked at it. He said, I don't know what this is. And if you look at it carefully, it's an orchid called Rhizoanthella. Apparently, it's pollinated by termites. Yeah. And the seeds are dispersed from a mammal, we think, a marsupial mouse of some type. Interesting. Then there's the favorite North American beauty, the ghost orchid of Florida and Cuba. And Cynorchus in Madagascar, you can't say no to that. That's a lovely terrestrial species. When I was there, I photographed this. There was a chameleon, you know, the real chameleons with the eyes that do this. It was the same color as the flower, and he was right there looking at me with a little beady eye. And I thought, that's really nice. So anyway. And then if you look carefully, there's even orchids that are on the ground in water, soggy water, like the spider orchid here in the Florida Panther Refuge. This is sometimes, you normally think a log in the water is an alligator, but this was not, and there was, you know, an orchid on it. Has a nice smell, by the way. It's very sweet at night. Platanthera is a very famous group of terrestrials here in North America. There's some right around Missouri here. In the Mediterranean, the bee orchids, they mimic wasps, and male bees attempt to mate with the flower, and they receive nothing. The flower gets pollen transferred. Odontoglossum crispum, the name's been changed, but this is one of the most lovely orchids according to several people I know. What do you think? You like it? Isn't that nice? Madagascar, though, I'd say is extremely interesting for orchids because they have 1,000 species on this island, the 4th largest island, 90% of which are endemic, found nowhere else in the world. Let's think about that. Of 1,000 orchid species in Madagascar, 90%, 900, found nowhere else. Problem is, 90% of these endemics are all highly vulnerable because Madagascar, unfortunately, has lost most of its forest. If you go there, they don't pretty much have any forest left in many areas. A lot of the pine trees that I was told were planted by the French are now used to cook with and other things. And so I would love to see what this was like 100 years ago. 90% of the natural vegetation is gone. And this is not just an isolated case in Madagascar and elsewhere. Madagascar is a snapshot into the future if we're not careful. We have a lot of invasive species. Every time I go to Florida, I see new species that I haven't seen before that are not native. Deforestation, poaching, and of course, climate change, the big one. And it's interesting that Pope Francis said that Madagascar is a lovely island rich in plant and animal biodiversity, yet this treasure is especially threatened by excessive deforestation from which some profit. The deforestation, the deterioration of that biodiversity compromises the future of the country and of the earth, our common home. That's the chameleon, by the way. And Ed Wilson from Harvard said, basically, if nothing is done about climate change, you can forget about biodiversity. My parents had property on the Gulf, and we watched the sea levels coming up inch by inch over the years, well, millimeters by millimeters, and there you go. It's not a good thing. And I was reminded of growing up as a child, this quote about the North American proverb, only when the last tree has been cut down, the last fish has been caught, the last stream poisoned, will we realize we cannot eat money. So here's a Mark Twain quote from nearby Hannibal. Mankind was not made for any useful purpose for the simple reason he hasn't served any. And that he was most likely not even made intentionally, and that by working his way up out of the oyster bed to his current position was probably a matter of surprise and regret to the Creator. I like that. So, but wait, there's good news now. This was just published a few months ago, about, I guess in October. Mike Fay at Kew and his colleagues wrote at the end of their paper, he said, there's now no technological reason why any orchid species should go extinct. Wow. We're at a crossroads. It might not all be bad. And an example of that was in a building about 100 yards from here. This is Rach Helmick. She's my former student. She was at the Missouri Botanical Garden here, along with Caleb Dvorak. And they came up with a technique. Now, Rachel is interesting. She does things in a very different way. She said, let's try an herbicide on this Platanthera from the Midwest to see if it will germinate better. And guess what? It did. How did that happen? Apparently, it keeps the abscisic acid from inhibiting the germination, and as long as you keep these plates in darkness, it'll work. Eventually, this herbicide, Floridone, will disappear. I don't have anything to offer. That's amazing. That's an example of technology, I think. So how do we do it? The Smithsonian, Dr. Dennis Wiggum, came up with this idea of this 4-piece puzzle. You have to fit these pieces together to make conservation of orchids happen. You need the fungi, because without the fungus in nature, the orchids can't germinate. You have to have something to pollinate these flowers to get the genes, to get the seeds with diversity. Then you have to learn how to germinate them. And then you can use that to propagate them and to release them back in the wild to help nature out a little bit. So here we go. This is I'll give you an exam at the end of the hour. All right, there's the endosperm that orchids don't have. Let's start with that. That's a refresher. The orchid flower. What is it that attracts the pollinators? For the most part, not all. The color, the smell. If you're flying around, you get closer to it, and then you might want to take a sip with your straw, if you're a butterfly or moth, into the nectar spur where the nectar is found. And this is what a lot of orchids use in nature. They try to reward pollinators more than they don't. There's a controversy that there's some saying that orchids never reward their pollinators. That's not actually accurate. Most of them do. And these are some of the moths found that pollinate this Platanthera integralabia from Kentucky using what we have called digital cameras. They actually activate when something flies around the flower, so you don't have to be there all the time. I did when I was in grad school. I had to sit there. But what can I say? The student that did, the PhD student did this is Tara Littlefield. She just set the cameras up and then she emails me and said, look at all the good flowers we found. So, all the nice pollinators. And then bees, of course, are important in the pollination of orchids. They're important as well. And we also have an ecosystem. We have the praying mantis shown here, and this is in Singapore, sitting on an orchid, devouring something that was trying to pollinate the orchid. So that's not being helpful. But the front legs grabbed it, and now it's eating it. By the way, you'll never guess what the close relative of the mantis is. It's the roach. Yeah. It's okay, they're mantids, they're not roaches. So this is a camera trap taken by a National Geographic photographer, Carlton Ward. And the top there explains that we don't know much about the pollinators. Why? Because nobody likes to sit in swamps 24 hours a day. But now we have a mechanism to do this. So I'm very excited about this. And this is another pollination story. Some of you know about Charles Darwin's orchid, Angraecum sesquipedale, the massive nectar spur. I mean, this thing is a monster. And Charles Darwin said, there must be a moth in Madagascar that has a straw-like mouth at least as long as this spur is to pollinate it. They've never found it until several years later. They found out that he was right. And so, you know, this is the scientist at work here. And I wanted to show you this picture because this is our pink-spotted hawk moth of North America. It's a big moth. It's lovely. There's its proboscis, its straw-like mouth. This is one of our biggest moths. There's a penny. This is Angraecum sesquipedale dried flower. And you can see that that's not going to cut it. It's not going to get into it's not going to make contact with the pollinia. It's a big moth, big. And back when I was in grad school, I used something called a camera with photographic film. And it was hard work back in the day. So we've shifted from old school photography, which, by the way, is lovely if you get it to do the right thing, to digital. And this picture was also taken by Carlton Ward. This was published in Florida Entomologist. I'm going to take you on a quick journey on what the camera traps have found with Florida's ghost orchid, the signature species of North America. You can't really deny that that looks kind of spooky. Little eyes, mouth, tongue. If you've never seen the ghost orchid in nature, I invite you to do so someday. Go into the swamps at sunset, alligator swamps, go about your belly button level, and just smell the air. You might hear a splash by you. It's probably just a pond apple, and that sometimes happens. But you'll smell it first, and then you look around, and it might be on the other side of that tree. And you lose all sense of time and space, and you forget the gators and all these other things. Try it. It happened to the British that we took there last year. They were like, yeah, wow. But anyway, not for everyone. This is one of the moths taken. This, I believe, is the first photograph of the ghost orchid being pollinated ever. There's another sphinx moth shows up. It's a really amazing. You can see the proboscis going into the flower. Butterflies by day came in, like the Polymedes swallowtail. It didn't pollinate it, but it was interested. And then a little geometer moth shows up. Poor little thing. There's no way that's going to pollinate. And another geometer, a little bit bigger. This is a green one that came in, and he's trying its best to get in there, but not going to happen. And then the biggest of them all, the giant hawk moth, did show up in the last images that they took, and it was attempting to go in there. You can see the proboscis and the length of the nectar spur side by side. This is one of the pollinators of the sphinx moths of this orchid.
Have any of the butterflies been successful in pollination?
Of the ghost orchid? Not to my knowledge. No, not yet. Yes.
Then there's less pollinators for it?
There is some speculation, or some people have proposed, that if you have a very long tongue, that you might not make contact with the pollinia because you're a nectar thief. And It's possible that some of these moths will do that, but in general, the best fit is going to get you to the spot where the pollinia are going to stick to your eyes usually, or the proboscis itself. I think I rounded about that question, answering that, but help me out.
It sounds like an arms race.
Yeah, yeah, that's what the Darwin orchid is like. You got to keep getting a longer mouth part over time, and vice versa. You have to keep it at, you have to keep at it. It's an arms race. That's a good way to look at it. One of my students went to the Panther Refuge in Florida. He did this. This was undergraduates, both of them. He said, let's take a turkey bag and put it on the flowers of a ghost orchid in the swamp and suck the air out with a vacuum pump hooked up to a battery. He did that. That's J.J. Sadler. And we sucked the air out, and we analyzed the chemicals in a carbon ampule, and we have the signature molecules of what makes the ghost orchid smell so good. The blend of these chemicals, we did this in the laboratory, we mixed them together in the right ratios, and it does have a very similar smell. So now we know what kind of scent the ghost orchid chemically releases. More technology. And we also are now looking at what kind of chemicals are in the nectar now. So think about it. When you go to when you go to some Chinese restaurants, there's one chemical that they often add to the dish. What is it? Huh, you said it, not me. This is an area that's not been looked at right now. And my colleague at Illinois College is a biochemist, and we're doing this right now. And there he is, Brent Chandler, and our student, Tony Ruiz, went to the Panther Refuge with a micropipette to extract the nectar. They took it back to the chemistry lab. They analyzed the chemicals in the nectar now, not just the scent. And there they are. Sugars, as you expect, is sweet. You can take the flower and, you got sugars. But there's alcohols. And why are they there? This is the ghost orchid's chemical signatures in its nectar. And I was told tonight that this would be a nice slide to show, so there you go. All the chemists in the room, there you go. And we went to the Royal Botanic Gardens in last May. Yes?
Question, has anybody ever looked at what the flower looks like outside the visual range of light?
Ah.
Is it a different color? Does it attract its pollinator by glowing in a range we can't see?
Yeah. Yeah, a lot of, apparently, some insects can perceive UV light. And they have these things called nectar guides. You're absolutely right. We're biased. We have different taste buds, smellers, ears, everything. We can't assume what we perceive is what they perceive. So I think you're absolutely right that certain orchids look and smell different to them than to us. For orchids that are pollinated at night, when you don't have a light issue, you have to be very visible. Something has to reflect the light to know that you're there. And the smell is going to be revved up so you can sort of get there. So yeah, that's what we're learning all the time. So we went to Kew Gardens, and we got permission to sample Darwin's orchid to look at the nectar chemistry. And we did see some specimens that Charles Darwin had signed himself. That was really nice to see. It turns out that the orchid of Darwin has chemicals that were sugars and something very similar to MSG. The paper's almost done. You can read about it maybe if it gets published. We're going to try. So that would explain why an orchid pollinator says, hey, this is really good. I might have a sticky thing on my eye, but I'm going to go to that flower now, and I don't care what's going to meet me there. I want more of that, So that's what we think. So back to the orchid biology. Obviously, up to 1 million seeds per capsule in some of these tropical orchids. Bob Dressler says you cannot discount the likelihood that 10 billion seeds could be blown from Africa to South America in viable condition. The ancestors of the ghost orchid, we think, were from African origin. And there they are, microscopic, usually. Different, they look different, but at the core, they have an embryo and a papery thin seed coat that's translucent for the most part. Dust-like, meaning that they can get blown into trees with the breeze to colonize the tree, 70% of which do that. If you look at them up close, you can stain them, and you can tell that they're alive, and you can store them in places like Kew at the Millennium Seed Bank. Orchid seeds do store quite well if you do it right, if you dry them thoroughly. There's Rachel. She's the one that worked at the Missouri Botanical Garden here. And this was when she was in my lab, and we wrote about her in the book. Basically, she came to the lab one day. She said, I'm going to take the seeds when you were a graduate student 30-some years ago out of the freezer, and I'm going to grow the orchid like you did at Clemson. And I said, don't even bother. It's not going to happen because they're too old. And she said, I'm going to use the fungus that you have in storage also. And I said, you're just wasting your time. And guess what? 30 years later, she did it. The seeds in storage in a common refrigerator, like the ones that we see in rooms like this. If you store them right, they can survive for decades, at least some. So how does this happen? The embryo, if you dry it thoroughly, you just keep it in a suspended animation state, and then when you rehydrate it, it swells a little bit, and it becomes receptive then to a fungus. The fungus comes in nature when a seed lands, bark or soil. The fungus, we think, sniffs around or detects that there's something good to eat here, to decompose maybe, and it goes into the suspensor region of the seed, of the embryo, and it infects the embryo. Now this sounds like doom and gloom. This orchid seed is toast. When there's a fungus infection, kiss it goodbye. Not so. This is the fungus now inside the embryo, one of the cells. It's called a peloton. The purple stuff here is the fungal tissue, basically. It's called hyphae, strings. And the orchids have learned over millions of years to digest them, which gives them the food they never were born with. This is the endosperm that they weren't born with. This is the carbon source that they're using now from the fungus because mom was too cheap to give it to Sorry, it wasn't very appropriate, I guess, but that's basically what, and let's not forget, if you're in the tree and it rains, it's not going to rain all day sometimes, so you got to have water. And fungi have a lot of water, so when you digest a fungus, you're going to get fed and you're going to get a drink. It's beautiful. There it is. There's a little monster. You take the orchid fungus, if it's the right fungus, you can actually get germination. These things are called protocorms. They're seedlings that don't quite have a leaf yet. So when you see this in the lab, a baby orchid seedling, this is going to be the root. This is going to be the leaf, the solar panel, to feed it through the sun. And the fungus is still there. That means that if a rabbit or something comes along, or a snail, and chops off the solar panel, it can still feed on the fungus in the root system. It doesn't go away. This is one of the most important things you should know about orchid biology. Orchids feed themselves two ways, not one like us, not like most plants that feed just with sunlight. They use the sun and they take energy from fungi together. They supplement their diet, depending on what they want to do. The problem is, for conservation, you can't leave the fungus alone. You got to get the fungus out and save it with the seeds. And that's what we do. And here's something I've been proposing at meetings. I want you to just think about this. Look at this a minute. Everybody knows caterpillars. When they're babies, well, when butterflies and moths are babies, they look nothing like the adult. Same thing with orchids. Protocorms kind of remind me of larvae. Now, calling a baby orchid a larva seems like not a very nice thing to do. But basically, it's a very similar thing when you look at these things. And it might be of interest to you that the orchid fungi that a lot of orchids like to eat or consume are related to chanterelles, and they make really good stir-fry dishes, you know. So I guess you can't blame the orchids for targeting the chanterelles for part of their nutrition. And this is what the fungus looks like up close in the laboratory. That's what they like a lot. So most orchids, I would say in North America and Ecuador especially, this is the genus Tulaznella. That's like the pizza that they all like. So it's very common. It's here in Missouri. There's another fungus that they do like as well. The ghost orchid likes this one. This is called Ceratobasidium. It's very aggressive. It's amazing that these orchids can tame this fungus and eat them, but this is one that, not one of my favorite to work with. So I want to show you, you know, the Wizard of Oz. Think about it. Who is being mean here? Is the fungus the enemy or the orchid? Who is taking and not giving back? Who's the wicked witch? The orchid is a taker for the most part, we think. Now, there's exceptions, but orchids don't appear to give much back to the fungus world. They take more than they give. And the orchids are, what can I say? Some people propose that they're parasites of fungi. And this is my former student who got his PhD and published this paper. This is another new technique. He has used some very high technology DNA sequencing to look for the fungus on tree bark in the panther refuge. You don't have to isolate the fungus from the tree bark. You can actually take a piece of bark and do the DNA work and you know what's in there. You can tell what kind of fungus is there. You can tell what kind of fungus is in the roots of the orchid just by doing the genetic work. It's fascinating. And how do you do it? From the old-fashioned way. This came out of a prairie in Illinois. That's Spiranthes. If you dig up the roots of a terrestrial here in Missouri, you're going to find this is bonkers. When I was in Madagascar with the British team, I was smelling these terrestrial orchid roots and breaking them, and the British said, "Larry, what are you doing that for?" I said, "They smell like Band-Aids." That's how I look for, well, smell for fungi in orchids. If you smell a fungus, a root that has especially these yellow regions, that's generally where we go to get the fungus out. And it smells like Band-Aids. I'm not kidding. And sure enough, they went to London with the samples that I had a subset of samples and we isolated together differently. They said, yeah, we got the fungi from those. It smelled like Band-Aids. And there's some more. I mean, this is where I would look. This is a Platanthera from the Midwest, orange. That word's got a lot, lots of roots. That one's got fungi. That would smell like a Band-Aid. I don't know why. There's another sneaky way. Do you know what that is? Young people don't. What is this structure? It's a 35, it used to be I'd give talks with these things. 35 millimeter slide mount. They're hard to find. If you take nylon mesh and you put it in a slide mount, sprinkle about 300 seeds of an orchid in there, close it up, staple it. The seeds are trapped inside there, but yet there's a porous membrane that the fungus can slip through, but the seeds can't get out. So if you put these in the ground and bury them for 1 to 3 years, you can fish for the fungus in nature and capture the little devil that starts the germination process. You can fish for fungi. We do this. We open them up, and there they are. This is Platanthera integralabia, or leucophea, excuse me. They're the little orchids that are trapped in there, and we got the fungus that did that. That's how you do it. Because sometimes these orchid fungi will shift, and they'll be different than when they start their lives. And this technique was pioneered by 2 of these very well-known authorities. Hanne Rasmussen and Dennis Wiggum published the paper that led to this technique. But Dr. Rasmussen, when she visited here from Denmark, she showed me how to look for baby orchids in nature without having to use the seed bank, the seed baiting. They're everywhere. She showed me how to look for them. I can do that now. I can look for orchids in trees. Small stuff. This was in Ecuador, Cuba, Florida. And once you get the roots that smell like Band-Aids, or if you find protocorms and you sacrifice this poor little beast, you get the fungus out, you chop it up with a sterile water droplet, scalpel, tweezers, petri dish, and then you chop it up and you add some agar to it, let it solidify. There are the root pieces. The fungus starts to grow out of them because it says, it's about time you rescued me. That orchid was being terrible. It was trying to eat my hyphae, you know, so I take them out. That fungus is good. It's good for the orchid. The orchid wants to eat it again. So we take the fungus, there's the peloton, this is the hyphae growing out of it under the microscope. All I do is under the microscope, take a sterile scalpel and take a piece of that, these hyphae, and then restart it as its own pure culture. And then you take that pure culture and you put it with the seeds in the laboratory and you have magic, if it's a match. And that's what it looks like. That's Tulasnella. Isn't that nice? Nice pure culture. It doesn't harden, it doesn't produce a lot of spores, it doesn't harm you. It's not like the mold in your drywall. Yes?
So do you have to then look at that from its genetics to figure out what species it is?
Yes.
Oh, it smells a certain way,
No.
it grows a certain way.
The genetics, thank goodness there's along now. Because when I was in graduate school, we didn't have that. But now we know that, for example, that Tulasnella is Calospora. We can do the genetics on it. We can see these things all over the world, actually. This is a very hot area of research right now. So the symbiotic germination, what my students and I do is we get the fungus and we pop it on a petri dish with orchid seeds, and if it's a match, they will germinate very quickly, very quickly. This particular orchid was grown from seed to flowering in Florida in 16 months using this technique. In fact, we doubled the population of the Spiranthes. We're releasing it in the wild. We were told that we doubled that population. There was one population left in Florida, and it flowered in 16 months. And the ghost orchid fungus, we do have it. There's a Serrata basidium. Dr. Kane, I showed you, he was a postdoc of Tom Sheehan's we talked about. That's his PhD student. We gave him the fungus from the ghost orchid, and it worked. The ghost orchid is propagated by Serrata basidium that they demonstrated at the University of Florida. So I'm going to give happy faces tonight to the puzzle about the ghost orchid conservation. We're all going to celebrate the ghost orchids. We understand it now more so than we ever did, and maybe we can actually save it from extinction because there's now just 900 plants left in Florida and 200 in Cuba. That's the current number. And one other thing you should know before we switch gears, Kingsley Dixon and Nigel Swartz said that basically the rarest orchids are the ones that want just one fungus, nothing else, or one pollinator, no more. They're specialists. If you're going to be finicky about your diet, and that fungus is the only thing you like to eat, then that might be why you're so rare, because maybe that fungus just isn't everywhere. And that's the problem with conservation. You have to figure out the orchid, and is it a specialist or not? And if it is, target them first, because they're the ones most likely to go extinct. And so for all the scientists in the room, I'm going to give you my spiel. You should make some effort to conserve what you study. The old days are out the door. No more fiddling around with nice things that are really interesting to know about, but that organism, if it dies from extinction under your watch, then you own it. Publish, but don't let what you study perish. Please, get with it. Now, back to the teaching and the public message. I'm happy to say that orchids are now internationally known. The plight of the orchid conservation dilemma. We have a obligation as orchid specialists now to talk to the world about it. They need our help. We are their voice. And so we're ambassadors. They are ambassadors for other life forms. And the Chelsea Flower Show is what we've been up to lately. We have been displaying orchid conservation displays there dating back to 2022 and 2023. This was our first display. We had the ghost orchid of Florida. We recreated the habitat at the Chelsea Flower Show. And it was a stressful time. Let me just say that we brought in from Chicago Botanic Garden about maybe one ghost orchid looked like it was going to open in time for the Chelsea Flower Show, just one. And we had it on the plane, cradling it gently, because if that one flower breaks off, I'm just going to get on a boat and go to the Caribbean island, and I'm not going to be around. So it turned out that during the week of Chelsea Week, the BBC showed up, and they interviewed me. I just happened to be there. They said, you need to go talk to the BBC. So the ghost orchid is behind me in the tree, and this guy, I was told to be careful of him because he likes to steer you a different direction. I was waiting for him, and I did. I steered him back. But he said, now, where's your ghost orchid? And I said, well, it's in bud. It's right here. And I had this crooked stick that looked like a Harry Potter thing. And I said, it's right over here. Here's the ghost orchid, and there's the bud. It's not opening yet, but give it some time. It might open by the last day of the show. And it was cold. We actually put heat pads around this thing at night to kind of keep the orchid thinking it's in Florida, not Great Britain. And so turns out that my wife was at the hotel. She saw the BBC article interview that night on prime time. She said, yeah, you're on there. Next day, I was sitting there. This is the ghost orchid, right, by the way, right there. Can't see, that's the one in bud. And Hugh Grant shows up. He said, I saw your interview. Can you show it? He has little girls with him. He said, can you show it to me? And I said, sure. And I had the Harry Potter stick, and everyone was asking about the Harry Potter stick. And the Spanish moss was a hit in London. They didn't know what that was. But it turns out that it didn't flower at the Chelsea Flower Show. It refused. Now wait, there's good news, because I wouldn't be talking about it now if it didn't. 2 days later, we took our ghost orchid as a gift to the Royal Botanic Gardens Kew, to the Princess of Wales Conservatory, where it was on time-lapse film. It was opening for the very first time. It turned out that the buildup was a good thing, because then when it did, it went boom internationally. This thing had 25 million social media hits when it opened at Kew. And it outranked the Ukraine war, I was told. And everyone in the world in the orchid conservation community said, congratulations, you really got the message out about rare orchids. And we were interviewed on, AP and everything like that. So this is how it began. And now we've been going back to Chelsea and showcasing orchids from different parts of the world. And last year we went to Chelsea. This is, they say, I took somebody to Chelsea and I said, well, it's in a tent. An 11-acre tent was where this all is all happening. And they thought it'd be a rickety tent. No, it's not. It's very lovely. And our display showcased the orchids of Hong Kong, Singapore, Hawaii, and Australia. And we took a team of scientists. My role was to invite the scientists and make sure they behave themselves, basically, and, and then put them at the Genius Bar to answer questions, because 150,000 people come here during the week, including very much dignitaries. So the University of Florida was represented by Carrie Reinhardt Adams here. That's Johanna Hutchins from the Chicago Botanic Garden, who brought the ghost orchid the year it flowered. And that's Ashley Clayton from Longwood Gardens. All of our logos are up, proudly displayed, all the people that we brought in. And this is what we do now annually. And this is what it looks like, the backdrop from last year. We have the Queen of Sheba from Australia, a lovely terrestrial orchid from Australia on the backdrop. The message is there. Orchid conservation is now looked at by people all over the world that go to Chelsea. And this is what it kind of looks like. If you really want a close look, we can take you behind here and show you things up close, but there is problems with people wanting to poach things even here. So we had to keep an eye on them. And this just shows you some of the orchids on display. Singapore. That shipment came in CITE permitted literally the day before this was taken. They finally got them through on DHL, came in. Very stressful. This is Hong Kong. It's neat because the team members from different regions all work well together from their perspective. We have to kind of make this all flow for the judging. Hawaii, I don't know if you can see this, but there's sand here, and one of our team members took their shoes off, and you can see the footprints that look like they lead down to the beach. So we talked about the native Hawaiian orchids in this particular display. The darling of the show last year was from Australia, the outback basically where Rhizanthella, the underground orchid, was, and also that's the orchid that got the gold medal for us. It's called Thylomitra, and it's a terrestrial. It's very blue, and it only opens when the temperature is warm enough. It wouldn't have opened when the ghost orchid was there, but it did just in time for the media to see it opening, and that's what garnered the attention. And we were visited by, there it is, when it opened, we were visited by Queen Camilla. She had a chance to see it. So, yeah, it worked out really well. We got a gold for that. We missed the gold by 1 point the year before, I was told. But, you know, it's amazing how it all kind of fit together, and the message was there. Yeah. So here's toward the end now. This kind of makes you feel good, I guess, me at least. It says, never doubt that a small group of thoughtful citizens can change the world. Education. Why are you teaching at a small college? Why wouldn't I? Look at that, Nelson Mandela. Most powerful weapon which you can use to change the world. Rachel, come on, she was in my class. Kennedy said, we choose to do this not because it's easy, but because it's hard. Because the challenge is one that we are willing to accept, one we are unwilling to postpone, and one which we intend to win. Wow. The co-author of Saving Orchids, Phil Seaton, he said some nice quotes. Conservation is all about people. Once people are aware of the problem, they want to help. They just need to know how. People can feel powerless, but there are actions they can take. So we wrote Saving Orchids, a book of stories to kind of encapsulate some of the positive stories in the world. And it's not all doom and gloom. Unfortunately, the shipment didn't get here today, but I have one copy you can look at if you wanted to buy it. We do have a QR code. If you like the Royal Botanic Gardens cover better, they co-publish it with Chicago. Some people like the ghost orchid on the British version. There's the QR code for that. So, our planet is essentially a massive greenhouse brimming with orchids and other creatures. Future generations will soon inherit this greenhouse from us, and they will look back on the actions that we take here today. What kind of a world do you want them to inherit? Come on. If you want to do something about it, the North American Orchid Conservation Center in the Smithsonian, there's the QR code. They're desperate for donations. You can help them out. There's a network of all these institutions that have a database, and we're all working to save the rarest orchids in North America from extinction. And with that, thank you.
We did have Caleb also come out and give us a talk here about philosophy.
Good. Yeah.
Repository of information
Yeah.
there.
Yeah.
Talk a lot about his mycorrhizal fungi. I'm not sure if he does that currently with a lot of his stuff, but I know he has some good techniques. That's who I use for philosophy.
Nice. Caleb is one of those people that he just has the knack. He knows. Nothing is impossible. Yeah. He has a business, I think, now. Any questions? And if not, I was very, very Based on the early work done with the molecular work to look into substrates, there are signatures of orchid fungi around. Now, is it that particular strain that one orchid relies on as a specialist? It's a hit or miss affair. Tules delicaspora is ubiquitous, but it might be patchy in certain places. So the orchid fungi as a group, not all orchids use this type of fungi, but many of them do, and they are mostly going to be decomposers. They don't really care about orchids. They just want to eat dead things, leaves, rotting leaves, and they're doing their thing. But the orchids have figured out, I'm going to take that fungus and take some energy from it, some carbon, and figure out how I can take and not give back, maybe. It's clever. I'm telling you, orchids, everyone in this room knows, they're master manipulators. Look at that wall. Where did the money come from? So, pollinators, fungi, they're part of us. We're being manipulated by them.
You mentioned that the fungal roots smell like Band-Aids. Is that a fresh Band-Aid or is that?
Yes, thankfully. Yes. Now, epiphytic orchids don't seem to do that. I've tried, but the fleshy terrestrial orchids here in the Midwest and Madagascar, I don't know what it is. Somebody should do that. I'm not kidding. Yes.
So you use these 35 millimeter slides. My father has about 600 or 700 boxes of them.
Wow.
Do you have a shipping address?
Pictures of them?
God.
I would actually like to offer the North American Orchid Conservation Center your shipment. I have plenty in my lab.
Okay.
Then, I'll be retired. I'm semi-retired in another 5 years maybe. But please, yes. I'll give you my card and I can put you in touch with them.
Every time we go down into the basement, I say, "What are you going to do with these?"
Yep.
And he says, "I don't know."
Yeah.
Cool.
I like the dumb things. They're great. Yeah. So you ready for your test?
I was just gonna say, how do you recommend to go about reaching out for? You mentioned that the rare ones are the ones that are specific to a particular fungi. How do you know about finding that? Do you have to search the literature? What's the best way you would recommend for people to kind of become interested and really champion a particular orchid to look into finding out which the ones that are?
Well, first of all, when I started my PhD work, there was one fungus I consistently kept getting out of the roots in the babies, the youngest stages and in the adult stages. So this one orchid seems to really like this one fungus throughout its life. Sure enough, we married them in vitro. They liked the fungus we gave it. And now that the genetic techniques are out, we can take the root pieces from all over the place and analyze them, and that fungus is there. So that's evidence that, backed up by the experimental germination, that this orchid really is a specialist. If you take the DNA, do the DNA work on what's out there with the roots, augmented by the lab stuff, you have a pretty good case that this is a specialist. And that's what I told the Fish and Wildlife Service. I said, this is an orchid that is a specialist, and they quoted me, and they got on the federally threatened list. So that was good. Yes?
You know how long the plant can be kept in cultivation before you really can't find the fungus anymore in its roots?
Depends on where it grows and how it started its life. The orchids in the greenhouses, I have one in my greenhouse, and they do have fungi from the log that I put it on. I don't know how it got there. So I would say it depends. If you're using a lot of fungicides in the greenhouse, I wouldn't give the orchid too much hope that the fungus is there. It's probably going to be mostly photosynthetic. And if the light goes out, then it's good luck. You got to get a fungus. So I think it depends on the species. Now, you remember the rhizoanthella from Australia? There's no sunlight in the ground. that's completely relying on the fungus as its carbon source because it doesn't have the solar panels to produce its own sugars. So that's the thing. Each orchid is its own unique story, and you had to figure it out.
Someone online asking about the orchid conservation that's going on with the endangered species here in Missouri specifically. You're obviously not too far away. Do you know if Mobot is still currently doing any of that sort of research, or anybody locally that is kind of specializing in the natives of this particular region?
I don't know the newest information now. I should know by tomorrow maybe if I go over there. But I know that Rachel and Caleb both worked there and were active in that area. And I have to say, I don't know because they were the ones I worked with, and maybe there's someone else there now. I'd like to meet them. I'm always available to learn from them, you know.
I can try and help you get in touch with that. We have people here that have a connection with Mobot too, but we do volunteer in the greenhouse history, so maybe we can get you connected.
That would be
If
great.
anybody else doesn't know, there's a research facility down Morgan Ford in the Tower Grove South neighborhood where they do a lot of this research. A lot of that work has been released off-site to give them the ability to do some of this stuff. I don't know if it's specifically native Missouri or.
Yeah, it's fascinating, and hopefully we've been inspired tonight a little bit. The orchids at Chelsea this year, we're going to be showcasing the orchids of China. Next year, we're planning to showcase the orchids of Bhutan. And that's the plan. And then it might be Florida and the Caribbean 3 years from now. That's the chatter. If you guys are in London in May, third week of May, and if you've never been to the Chelsea Flower Show, it's an experience, to put it mildly. It really should be seen to be believed, because even the slightest spot on a leaf, you're out, you know. Yes?
One other question. We've talked a little bit about displays. I like to put up displays. You have beautiful native displays. Do you guys go about really planning that to get native plants in there and really kind of map out that whole system, or just bring stuff in and see how it fits in there?
Yeah, I neglected to tell you about my partner at Chelsea. It's John Park Wright IV from Florida. He's a businessman, I'm a scientist, and we kind of spearheaded this together. He's a wonderful, wonderful person and a friend. And we're just trying to inspire the conservation in a public area, and it seems to be working. And I hope it continues. And I think that it would be well worth to go see this at some point while we're still active doing it, you know. And to answer your question, this week we usually have one Zoom meeting leading up to May, and we had to have all the specialists on the Zoom, and we have a general idea of how it's going to look and what it's going to say, and it's nice what we have planned this year. I'm not telling you about it yet, but it's amazing how we have to have, and there's sometimes arguments. It's like, no, you can't do that. So you have to balance the education and the conservation with the judging, and sometimes it's mixing oil with water, and you got eggshells. You got to walk around the eggshells sometimes, but it all works, and I don't know how. Yeah.
For the Western terrestrial orchids, they always say, don't take them out of the ground because it's going to die.
Mm-hmm.
That's always,
Yep.
nature, don't poach them. Is it because the fungus dies and the plant dies?
Quite possibly. They have connections sometimes with trees. Sometimes, for example, Isotria seems to link with the Russula mushrooms that link with the tree. And if you sever the connection, good luck getting it to restart again. So that's the kind of thing we're dealing with terrestrials, especially in the far north. They're very fussy about being moved at all. If you're going to try this, I don't recommend it, but start with the little stuff and recreate. Maybe take the soil with the fungus in it and mix it in. The fungus has its mind of its own if it's going to like that or not. And if it doesn't, then it's off it goes. So it's hard to play God and nature in your backyard and get it right with them. Epiphytes, that's a little different. Good stuff.
Thank you guys very much.
Thank you.
QR code for the book. Do you know if that links to your other one, if anybody's interested?
If you want the Lepanthes photograph,
That's that one? Okay.
hang on.
Otherwise, it's in our newsletter too, the link to the website to pull up and order the book as well.
That's the,
Copy you can check out.
it's the exact, I got the I've got the Lepanthes over here. I can show you that book. We can pass around, or you can look at it. It's a show. It's for you guys to look at. It's getting good reviews right now, which kind of surprises me. But this is the Lepanthes picture. This is the U.S. version.
Thank you guys.
I'll leave it on the desk. You guys can look at it. I do have this copy if anybody wants to buy it. We offer it for $5 less than the publisher does because it's the author copy. So, but this is it. And if you don't like the Lepanthes, then you get, it's the same book. This is the one from England, the Royal Botanic Gardens, has our ghost orchid. I've been told they like that a lot. A lot of people do. Is there a show of hands? I can tell the publishers which they like. How many of you like that one better than the other? How about the other one?
Show that again.
How many of you like this one better?
I do like that.
How many of you like that better? What do you think? You're the guy that has to tell me.
That was about an even split, so it's probably.
Yeah, that's cool.
Half and half.
Yeah, that's cool. Yeah. So, there it is. I think it can work.
Thank you again.
You're welcome.